and welcome to Ask Your Academic. Today's session will cover the MSc Immunology and Inflammatory Disease, and we're joined by our programme lead who will do the best to answer all the questions that you have. So please do use the chat box to ask any further questions, and we've also got some questions that were submitted beforehand that we'll run through too. So Kevin, we'll pass to yourself. Could you introduce yourself and tell us a bit about the MSc, please?
Hi everyone, my name's Kevin Malloy. I'm a professor of immunology at the School of Infection and Immunity, and our school hosts the master's programme in Immunology and Inflammatory Disease, or what we shorten to IID. I'm a mucosal immunologist, so my research focuses on the intestine and how our immune system interacts with all the foreign molecules and microbes, most of which are beneficial in our guts, like food and commensal bacteria, but sometimes we get pathogens in our gut. And I'm interested in understanding how the immune system achieves this balance between being tolerant of the beneficial foreign things in our gut, but responding to any dangerous ones that come along. So the program that we run is a 1 year, primarily taught master's course. Uh, as I said, it's the immunology and inflammatory disease. And the reason it's called immunology and inflammatory disease is that our department has a lot of specialists in areas of inflammatory disease. So our course is a big translational science component, especially around of obviously chronic inflammatory diseases. You might have heard things like rheumatoid arthritis, the big strength, but also other musculoskeletal disorders, things like inflammatory bowel disease, which is something that my research intersects with. So the course is a 180 credit course, and it's taught in 3 semesters. And the first semester is really some background knowledge. These are compulsory courses. One of these is the basic and translational immunology. So there are a series of lecture sessions there that continue throughout the first semester. And that's to make sure that everyone, because a lot of people come at this degree from different perspectives, from different areas of expertise. So we start really quite basic immunology and build up to quite a high level. So the idea of this main course, which is a 40 credit course, is to give you all a very broad and deep knowledge of the key topic of immunology, and then in the context of inflammatory disease. So the lecture course kind of starts with basic immunology, and the last part of the lecture course of the first semester talks about various inflammatory diseases and gives you examples, and they're all taught usually by experts who do research in that field. The other thing that you do in the first semester is another compulsory course called Research Skills, and that's all about teaching you more practical techniques to understand these, and also you do a few little practical labs in that and get assessed on those as well, and that's 20 credits. So that's the first semester, all compulsory, 60 credits. And the second semester is where people can choose more what they want. We have 60 credits, but most of these, or 40 of these, are optional courses. So for the first part of the second semester, so that's usually from the January onwards, we have little blocks. There are the first, you choose 10 credit courses. I was sort of looking for these. Just give me 1 second. We have a lot of these courses, so I just wanted to see a little bit about yeah, I had it open a second ago. Sorry, give me 1 second. I need to open it again. So you can choose these. I'm only looking at this now because there's quite a lot of different courses, and we've increased the breadth of these courses recently. So in the first part of the second semester, we call that Block 2A. You choose 10 credit courses, and these would be things like I can see emerging viruses, that's very topical, neuroinflammation, single cell and spatial omics. So there are a lot of both different subjects and different technologies that are covered. And then you pick 2 of those courses for Block 2A, and we have something like 7, I can see, 6 or 7 different ones. And then in Block 2B, you do a 20 credit course. And again, we have different ones. A lot of these are in response to to technical advances that students wanted to find out more about. So one of these is about bioimaging for research. Another one is about tumor biology, and another one is about um, understanding omic data sets and using statistics and R for bioinformatic analysis. So you choose one of those, and that's 20 credits. And the last part of the second semester is what we call designing a research project. And so this is where you um, start to gain an understanding of what it is to actually do some hands-on research and to set you up for the third semester, which is really just your research project. So you get 60 credits in the second semester as well. And then the final semester is really just doing your research project. And that gives you a chance to go into one of these labs and to spend usually 10 to 12 weeks in the lab working on a project. And you have to write up a report at the end of that time. You have to present your work and get assessed on that. And that's the last part. That's the last 60 credits. And so you have 60 credits per semester, which overall gives you 180 credits, which is what builds up our master's degree. And that's quite a lot to take in. So I would be very happy to answer any questions that people might have about that. I think that's covers that question anyway. Yeah,
Yeah, no,
it's an
I think
overview.
that was a really good explanation. What kind of background do students on the MSc typically have?
We have lots of different backgrounds. So you will see on the university application website people with relevant degrees, usually around the level of a 2-1, so an upper second class, but sometimes a 2-2 we also take. In biomedical sciences, that could be anything. That could be pharmacology, it could be microbiology, it could be biochemistry. It's often there are a list of the kind of courses that we would, health sciences, things like that. There are a list of the courses that we automatically consider to be appropriate for those that want to study immunology and inflammatory disease. And then there are other what we call non-conventional routes. So, for example, if you have a background in veterinary medicine or even in medicine itself, we often have applicants from those fields. And what happens with those ones is I get sent these applications that don't automatically qualify for the entry criteria for the course. And it does, again, I often get people who have done different unusual degrees. Um, and so we don't discount anybody. Every single application that isn't automatically accepted or rejected on the basis of academic grounds gets sent to me. And then I can evaluate the candidate. I look at their academic track record. I don't worry too much about whether they've had any prior knowledge of immunology or not. I look more at their evidence that they are a strong student, that they're determined to learn, and that they work hard. They've got a good work ethic. And then I can then decide whether to accept or not to accept these students, and if there are any specific conditions associated with that. Sometimes that will happen. So if you haven't finished your course already, you might get what we call a conditional offer, or if you haven't got a certificate that your English level is at a high enough level that we require, again, you might get a conditional offer, which means that you know you've got an offer there if you make these academic levels on your current course and or the English requirements through taking one of these English assessment courses. Yeah, so there's not one background. We have a diverse set of students, various backgrounds, and we're pretty broad in terms of the expertise and experience that we consider across the student body that will accept. If you're a good student and you're clearly interested in learning more about this, then we're usually very happy to have you.
Great. And you mentioned the optional courses for semester two. When
Yeah.
is it students are expected to choose them? Is that semester one near the end of it, or is that right at registration?
No, so what happens towards the end of semester when we have an information day where I should say that every sub course, so we've got the course, the overall course, which is the master's course, but I said it was split into 180 credits, and then each course has a certain number of credits attached to it. And each one of those sub courses has a course leader. And for those optional ones at the beginning of the second semester, um, what happens then is that towards the end of the first semester, we have an information day where all of those course leaders come and present give an introduction to the course, what it's about, give you an overview of what it's about, and then you can make an informed choice on the specific options that most interest you.
Perfect. And one of the questions we got was about the project. So it was, how are students assigned a research project and can they pursue their own research interests?
Okay, so I guess the answer to this is yes and no. So it's a kind of a mixture. We normally have about 25 to 30 students, and we have an equivalent number of projects. If we get more students, then it can be a bit harder finding extra projects. I need to go and ask people to post a few more. But in general, what happens is, so let's say there are 25 students and we have 25 academics that have submitted projects. What happens then is we circulate a list of the projects and supervisors to the students and ask them to choose their top 5 out of those 25. And they submit the top 5, and then the person who runs the project part of the course, that's considered one of the sub-courses, the project, the research project, they use an algorithm which ensures that everybody gets their highest ranking choice as far as possible. So nearly everybody gets either their first or second choice of project. Occasionally it might be third or fourth choice, but you do have an input into that. Very occasionally there might be a student who has been very impressed by a particular lecture or session that we've had in teaching and has contacted that lecturer subsequently to ask about projects. And that can occasionally happen that that person will agree to submit an extra project specifically for that student. So we don't actively encourage all the students to do that. It would be a bit chaotic, but if there are students who are particularly taken by one aspect of the curriculum that they've been taught, and they feel that that person would be a great person to work with, and that person's happy to take them, so there's a lot of ifs here, then sometimes they can kind of arrange their own project. But that's very much a minority. In most cases, the students select their top 5, and they're then allocated one of those through the algorithm. That's the usual way. And just to say that not all the projects are lab projects. So especially nowadays, so much of biomedical sciences is what we would call these kind of omics type technologies and bioinformatics analysis. So we've increased in our course in the last few years, we've increased the provision of teaching across these topics within the course to be able to go a bit deeper into these techniques. And so as a consequence of that, we also have started to offer what we call dry projects, where you're not actually in a lab doing experiments, but you're looking at data sets, either publicly available data sets from patient cohorts, for example, or data sets that our investigators have generated, and they want someone to do some analysis to look at specific questions or correlations of the data to try and formulate hypotheses. So that's getting more and more popular. I'd say it was still a minor proportion, maybe a quarter to a third of our projects are what we would call dry projects, where you're not doing the experiments, but you're doing the analysis. And there's also an option to do a dissertation where you really write about a summary of what's known about a certain topic and investigate that. And that's another one. There are a few, 2 or 3 of those that are offered and taken up every year. So for the people who really are not that interested in working inside a lab, there are alternatives that they can do. But the majority of our students, about 3/4, I would say, to 80%, end up working in a lab for their project.
Perfect. And what sort of lab skills might a student develop?
Well, what we hope is that they'll develop some practical skills. So depending on the lab that you're in, it might be more of a cellular immunology lab. Maybe you'll learn about cell culture. Maybe you'll learn about purifying different immune cells, the different types of assays that we do to measure maybe the responses to different stimuli and what things they make, and how we can evaluate their activation status and things like that. So there's a lot of technology in that respect. Other projects might be more molecular, where you're looking at gene expression within cells in response to different conditions, or purified from different tissues, or again, data sets that have been generated. So that's the second sort of main thing. I would say cellular and molecular techniques are very, very common. Often a combination of both of those. You might have some microbiological techniques if you're working on a project that involves a kind of infectious agent. You'll be learning about culture of maybe bacteria or viruses and using those to stimulate cells and looking at the response. So you learn some basic microbiology techniques as well. It really depends a little bit on the project you select. And lastly, there are all these informatics skills that we're teaching more and more, and these are getting applied not only to the dry lab projects where you get a data set, but often even when you generate some data yourself, you can apply some of these bioinformatic techniques, so computing skills and so on. So there's lots of, and the last thing that we want you to do in terms of the project is we want you to develop analytical skills. So you start off learning the technical aspects of the project, but then by the end of the project, you should really be in command of it. You should understand how to design experiments to assess a particular hypothesis, how to interpret the results. So you need to understand the technology. You need to be able to produce graphical representations or otherwise summaries of your results. Discuss that. And then from those, formulate next step experiments that bring us a bit closer to the aims of the project. So we really want to teach people technical skills, but also analytical skills as well.
Yeah, it's such a big bit of work, isn't it? And in terms of the teaching location, whereabouts are classes typically held?
So, so timetabling is done centrally now in the university, which means that we have the timetable is pretty much set, but the university's actually been building a lot of new teaching resources in the last, I would say, 5 to 10 years. And so the timetabling that's done centrally, we don't have one specific room that you're in most of the time. There are various teaching buildings, and depending on the size of the class, some rooms are suitable for up to 30 students, some rooms are suitable for up to 50, some are up to 400. So it depends a little bit on the size of a particular cohort. But in general, most of our lectures are around the central campus of the university. Well, they're never really off the main campus. But you can be at either end of the campus, but it's probably about a 15-minute walk to go from one end to the other. So yeah, there are various rooms. You kind of learn your way around the campus quite quickly. There's a little app that we have called University of Glasgow Life app. And when you register and you're here as a student, you download that app onto your phone and you click on that app and it will tell you where your lectures are. And it has little maps to tell you exactly how to find all these rooms. In fact, I have to use that to find the rooms myself sometimes, and most of the lectures are like that as well. So we all use that app and it gets us where we need to be when we need to be there.
Perfect. The beauty of old buildings, isn't it? It's not always doesn't make
Yeah.
always make sense where things would be. And I know this is a really difficult question to answer, but we're always asked it. Like a typical week, I know there's no such thing, that will vary from week to week, from semester to semester. But how many hours a week should students expect a contact time versus what's expected of them out with for self-study?
Yeah. So in the first semester when we do the lecture courses, you probably have 3 or 4 lectures a week. Now, the course is designed, and this is what separates a master's course from most undergraduate courses that you might have been exposed to already. Master's courses are designed to promote self-directed learning. So for every hour of contact with staff, so every 1 hour lecture, you're expected to put in about 10 hours of independent work to understand, revise that topic, and do some deeper reading. And you'll be given reading lists and so on to help you do that. So we can't have 20 lectures a week because then you'd be having to spend 200 hours a week trying to catch up on that. So normally for the lecture part, we have about 3 or 4 lectures a week in the first semester. Um, and then, but you're meant to do a lot of self-directed learning. Also, we have um, in parallel, we have the research skills course that runs also in the first semester. But again, that's usually, I think, 1 or 2 afternoons a week for part of the semester. So most of the time you won't be in an organised teaching activity when you're here, but you're expected to be using that time to build your knowledge, to revise on what you've been taught, and to prepare for anything that's upcoming. In the research project, that's full time. So you're expected to be in the lab full time, 5 days a week, depending. They won't ask you to work weekends or beyond normal hours. And you probably most people don't spend 5 hours a week at the bench doing experiments 5 days a week. Usually you're doing a few experiments a week, but again, there's lots of analysis, reading that you have to do around a project, processing your data, generating outputs, and so on. But essentially, that's full time. So for that last part, you kind of we want to give people the idea of what it's like to work in a lab full time. We want to show you that. So it does vary a little bit, but the first semester, lots of self-directed learning. Second semester, almost the same. You probably have a little bit more course, taught course for every one of these optional courses. You probably have a lecture a day almost, but they only last a couple of weeks. But again, lots of self-directed learning to build up your knowledge of that. Yeah, I think I guess the time that you spend in class gets bigger as you go through the course.
Perfect. And what about assessments? What sort of assessments can students expect?
We have lots of different assessments. Each course is assessed in a different way. And the course assessments are sort of formulated to see whether or not you have acquired the skills or achieved the intended learning objectives associated with those courses. Um, the main course that we run in the first semester, the basic immunology knowledge, we have 3 exams at the end of the term. I should have said this before, but one of those is just an essay exam where you sit in an exam hall and you're given an essay paper and you have to write 3 essays in the space of 2 hours. And you have a choice. Normally we have 5 topics and you write essays on 3 of those. That's really to see whether or not you've demonstrated understanding of the core immunology and inflammatory disease knowledge. We also have an exam related to that called critical analysis of a research paper, because again, something beyond undergraduate work is that we want you to read primary literature and be able to understand it. It's very important for your research project too, that you can go and read papers and scientific articles and understand them. So we're building that knowledge up through the first semester, and we give everybody this paper. We give you a couple of days to read the paper, and then you, again, under exam conditions, you have a couple of hours to answer a set of questions related to the paper. And the third thing that we do I should have said this, the third thing we do for that main course, the 60 credit course on basic and translational immunology, is that in December, in the beginning of December, we take the whole class to the British Society of Immunology annual congress. And this is great because then you get to go to a real scientific congress. You get to see world leaders present their science. You get to see also there are poster sessions where there are PhD students presenting their science, and you can go and chat to them about that, what it's like to be a PhD student. And there's usually about 1,800 people go to that conference. So it's the biggest immunology conference in the UK. And that happens always in the first week of December. So we take the students there. It's all part of the course. Usually it's in Liverpool, although the year coming up, it's actually in Glasgow. So we won't be going away for it. But normally it's in Liverpool. But we go there, and as an assessment related to that, we ask you all to write a dissertation. And the dissertation is about one of the research topics that's presented at that Congress. So the Congress is divided into different sessions. And what I do is I select 4 or 5 of these sessions, and the students again are allowed to select the top 3, and you're allocated one of your top 3 topics, and you're expected to attend that session. You're expected to read around the work that's been done by the people that present at that session. These are international leaders in the field usually. And then you have to submit a dissertation by the beginning of the next semester, so by January. So normally people do that over the December break. You have about a month or so to write this dissertation. You don't do it completely on your own. You have a mentor Each topic will have a mentor. That mentor will usually be one of our academics who's attending the Congress and will be at that session. And they will meet with the group of students. There's usually 5 or 6 students doing that topic. They'll meet with those students as a group and talk about the students' ideas of the topic that they want to write the dissertation around. So everybody always has a slightly different take on it, because each session will have maybe, anything between 3 and 6 different speakers. And so they'll all be talking about slightly different things. And that's a very important thing, that BSI dissertation, because it's different skill set. So it's really about assimilating information that's presented at the Congress, but also in combination with going back to the primary literature and looking through what's known about that topic and formulating some kind of summative summary of the current knowledge of that field. And that dissertation actually counts for 50% of your grade for that course. So that I told you that course, or the basic immunology course, was 60 credits, but actually 30 of those credits are just given by the BSI dissertation. Our students love attending this Congress because they really feel that they're hearing about cutting edge research and new things in immunology. And it gives them really an opportunity to see a bit further down an academic career pathway, what sort of things you would be doing. Not every week you go to Congress, but this is where you network, this is where you hear about new things, and this is where you present your findings, get feedback from the wider community. So that's that. Other assessments, like in the second term, at the end of every block, every optional course, there's an assessment that's linked to the material that was presented in that course. They take different formats. They're often submitted online, so they're not sat under time restricted conditions in an exam hall, but you do that in your own time. You'll have a deadline by which time you can submit that assessment. And again, those run through the semester. And for the research project, we have one part, the designing a research project, which you do in the second semester. When you've already selected your project, you go away and read about the project, and you make a poster. So you make a scientific poster the same way that students and postdocs do when they go to a congress, and you make a poster about your project, what it's going to be about, what are the techniques you're going to use, what's the question you're trying to ask. And we have a poster day where there are a couple of examiners come and look at your poster, and they ask you some questions about it, and you get graded on the poster itself, but also how you dealt with the questions, how well you can explain it. And then the big research project, when you actually do it, you submit a report at the end of the project, and we also have a day of presentations where you present a summary, and both of these get graded to give you your overall grade for the research project, which is the final 60 credits. I think I've covered most of that assessment.
Yeah, no, that was great. And you kind of touched on this, but what sort of things do graduates of the MSc go on to do?
We have people doing all sorts of things. Quite a few go on and do a PhD. So there are probably about, it's not the majority by any means, but quite a few, maybe about a fifth to a quarter, end up getting accepted in PhD programs. So the qualification that you get, because we also grade the students, so you can pass the masters, but if you do a little bit better, you can get a merit. And if you do very well, you can get a distinction. So quite often the ones who get distinctions or high merits, they often can get the chance to go on and do a PhD. Others go into companies, work in biotech companies and so on. Many go back to the place that they were before. They've got this additional qualification. They might be working in a health authority or a hospital or some other educational environment or health environment, and having the qualification helps them progress up. And some become clinical scientists and so on. And we don't have the data in yet, but I suspect that more of our students who acquire these analytical skills associated with bioinformatics. So these are kind of quite high level computing and mathematical skills now. And these are very much in demand across all sectors. So within health sciences, or biomedical sciences, but also just in every walk of life now. So we haven't been following these students long enough to know where they end up, but I suspect they'll end up in all kind of businesses and using these transferable skills to advance their careers that way.
Perfect. And then in terms of preparing for the MSc, is there anything students can do before they start their studies in September to set themselves up for a good start?
Yeah, if you get accepted, normally what we do is we send you some recommended reading before you start the course. And usually I do an introductory session right at the very beginning of the course where I give a bit of an overview of immunology and I give some basic stuff, but I also explain where we think you'll be at as you start and where we want you to get to by the end of the first semester to have a solid understanding of it. But yeah, we'll give you a reading list and we'll give you, for all the lectures, we give you handouts and so on in advance of the lecture, so you can even go do a bit of revising before that. But in ahead of the course, there's a little bit of background introductory reading to immunology that you can do. It doesn't have to be so deep, but it helps if you get some basic understanding of the immune system and you know the names of some different cells that are involved. But we send you that you can do that.
Perfect. And just get here nice and early as well. Get set up all that life admin elements, isn't it? Like finding our bank accounts, things like that. And then you can
Yeah,
just concentrate on your studies.
yeah.
Um, well, perfect. That's us covered all the questions. There's been loads of information shared there. So please don't panic if you think of a question afterwards or you'd like a wee bit more information on anything. We do have our email address on the programme webpage to get in touch with us to ask those questions. The programme team are brilliant at getting back to people. So yeah, don't panic. We will send the recording just shortly after this. And yeah, we'll hopefully see you all come September. So thanks, Kevin, for your time. And yeah, we'll see you all soon. Thank you. Bye bye.
Okay. Thanks, Naomi. Bye, everyone.